Welcome to this explainer. Today we're exploring one of the absolute most fascinating, kind of mind-bending paradoxes in all of biology. Get this. The exact same microscopic mechanism that's responsible for creating new life and healing your damaged tissue, it's the very same mechanism that can cause devastating malignancies and drug-resistant cancers. It's this incredibly delicate, high-stakes balancing act happening inside of us right now. And, well, by the end of our time together today, you're going to understand exactly how it all works. Okay, let's dive into this. Here's our roadmap. We'll start with the spark of life, look at the mechanics of cell merging, and see how we're measuring the invisible. Then, things get intense in the survival funnel. We'll uncover the risks in regenerative medicine, and finally wrap up with the dual-edged sword. All right, Section 1. The spark of life, a natural physiological mechanism. So, cell fusion and tunneling nanotubes aren't just weird laboratory anomalies. They actually occur continuously in perfectly healthy bodies. It literally starts at the very beginning, with conception, the foundational merging of sperm and ovum to kick off embryonic development. But it doesn't stop there. During gestation, tissue formation, and placental integration rely heavily on these fusions. And even, right now, sitting there in your own body, your specialized somatic cells, neurons, and immune cells like macrophages are constantly interacting and merging to keep everything running smoothly. It's just a continuous, totally natural physiological mechanism. Moving on to Section 2, Mechanics of Cell Merging, Connecting the Dots. Now you might be wondering, how do these microscopic entities actually communicate and merge? To bridge the gap, biology utilizes tunneling nanotubes, or TNTs for short, and this brilliantly illustrates that process. Notice that striking cobalt blue filament stretching between the green and red cells? That is the physical biological scaffolding connecting them. And those goldenrod yellow clusters? Those are essentially data packets. They act as molecular pathways, integrating precise signals and cellular machinery from one cell directly into another. They're literally sharing the basic building blocks of life across this delicate bridge. These architectural blueprints reveal just how microscopic this whole process really is. We're talking about a physical gap that is often less than 1 nanometer. But, these connections aren't all built exactly the same way. The models vary quite a bit. You have thick tubes for transferring entire massive organelles like mitochondria, and then super thin tubes just for tiny molecular signaling. There's lateral interlocking of membranes, and even this concave invagination, where one cell physically accepts the protrusion of another. It's an incredibly sophisticated structural system. Now, just to keep things crystal clear, we need to separate these exchange methods out. Tunneling nanotubes are essentially temporary biological scaffolding across a tiny physical gap. Direct cell fusion, on the other hand, is a permanent membrane merging where literally zero gap exists. The entire cytoplasm, nuclei, and chromosomes are completely combined. And lastly, phagocytosis involves the complete physical engulfment of extracellular material, or even foreign bacterial plasmid. Understanding the huge difference between a temporary TNT bridge and a permanent cell fusion is absolutely crucial for what we're going to talk about next. Which brings us to Section 3, Measuring the Invisible, Tracking Fusion over time. So, tracking these invisible interactions, it's taken about 5 decades of incredible scientific progress. Back in 1971, researchers were inducing cell fusion using the Sendai virus, and they tracked it manually. I mean, literally sitting in analog dark rooms, physically counting radioactive exposure dots on X-ray film plates. Crazy, right? Contrast that with today. We now use automated, high-throughput fluorescent monitoring and long-read next-generation sequencing. This lets us map natural fusion continuously, in real time. To track this today in the lab, we use an in vitro flow cytometry assay. Basically, we start by inputting 2 sets of cells. Stem cells engineered to express a green fluorescent protein, and another set engineered to express a red fluorescent protein. After letting them co-culture together for 24 hours, they are run through the flow cytometer. And the output is just incredible. Alongside our distinct green cells and distinct red cells, we see a totally new quadrant of yellow cells. Those yellow cells are our definitive, visual proof of direct cell fusion. But, proving it in a petri dish just isn't enough. We really need to know if this happens inside living tissue. So, in a brilliant in vivo experiment, researchers injected red fluorescent breast cancer cells alongside green fluorescent mesenchymal stem cells into a subcutaneous pocket of a living mouse. Over a timeline of 14 to 51 days, they carefully analyzed the tissue. And once again, what did they find? Those same yellow fused cells. This verified beyond a shadow of a doubt that cellular merging happens continuously inside complex, living biological systems. So, the absolutely crucial point here is the specific fusion rate we're seeing in the data, between 0.1% and 1.0%. Now, at first glance, a fusion rate of less than 1% seems incredibly tiny, right? Almost negligible. But biologically speaking, when you consider the billions and billions of cells in a human body, this microscopic percentage actually represents a massive, game-changing evolutionary event. Let's look at exactly what happens next in Section 4, the survival funnel, evolution, and chaos. To really understand the chaos that ensues, we first have to understand karyogamy. This is the actual fusion of the cell nuclei, and it results in some dramatic, chaotic chromosomal shuffling and entirely unpredictable genetic recombination. During karyogamy, a cytoplasmic merger initially creates this dual nuclei state. But then, the nuclear envelopes completely dissolve, and the chromosomes literally shuffle together into one chaotic mix. The outcome here is highly volatile. While some cells temporarily fuse and then just split back apart, others face uneven segregation. This basically means they divide with the wrong number of chromosomes, which leads directly to deep, dangerous chromosomal instability. Now, what's really interesting about this slide is how it creates this harsh cellular survival funnel. If we start with, say, 10,000 baseline cells, only a tiny fraction actually initiate fusion. Because of the sheer chaos of that uneven chromosome segregation we just talked about, 99% of those fused cells simply stagnate or they trigger apoptosis, which just means they die off. However, that incredibly microscopic fraction that does survive, it adapts, and it emerges at the bottom of the funnel as a highly robust, unregulated, and potentially malignant supercell. Section 5. Risks in regenerative medicine, the unintended consequences. Okay, so one of the absolute most terrifying capabilities of these fused supercells is acquired resistance. When a vulnerable cancer cell fuses with a resistant cell, it doesn't need to slowly mutate over generations to survive chemotherapy. Nope. It directly inherits fully functional resistance machinery, specifically these specialized membrane pumps. So when clinical therapy drugs attempt to penetrate the cell wall, this newly acquired receptor mechanism physically captures the compound and forcibly ejects the drug straight back out into the extracellular space. It literally guarantees the tumor's survival. This creates a really stark divergence in cellular control. Normal stem cells undergo strictly controlled differentiation and very balanced proliferation down specific lineages. They listen to the body's signals and they accept triggers for cell death when they're too damaged. Cancer stem cells, however, the ones born from these malignant fusions or unintended recombinations, they are highly erratic. They feature aggressive, totally unchecked duplication. And worst of all, they actively ignore the body's structural signals, effectively evading cell death altogether. And here is the massive why this matters moment for the future of healthcare. Regenerative medicine heavily relies on injecting therapeutic stem cells to heal damaged tissues. But there is a huge hidden risk here. These injected stem cells have an inherently high capacity for differentiation. When you place them in physical proximity to host tissue, the local environment naturally triggers the formation of those tunneling nanotubes we discussed earlier. The collision of these high-capacity therapeutic cells with host tissue creates a highly unpredictable environment for unregulated genetic recombination. So, in our pursuit to heal, we might accidentally be rolling the dice on unintended malignancy. Finally, Section 6, the dual-edged sword, a biological paradox. It really all comes down to this. The exact mechanism required to generate life is identically responsible for enabling malignancies to survive it. It is a true dual-edged sword. Following the adaptive path, natural cellular fusion yields positive evolutionary survival, the absolute miracle of fertilization, and essential tissue repair. But following that exact same cellular fusion mechanism down the malignant path, that leads to chromosomal instability, unpredictable genetic recombination, tumorigenesis, and an unyielding therapeutic resistance. It is literally the very definition of a paradox. Which leaves us with this final, really profound question to consider. As regenerative medicine advances and we keep pushing the boundaries of science to cure disease, can we learn to harness the incredible healing power of stem cells without accidentally unlocking the chaos of uncontrollable cellular evolution? It's a delicate, incredible balance encoded into our very biology, and decoding it is going to dictate the future of human health. Thank you so much for joining me for this explainer. I hope this really helped you visualize the incredible mechanics happening right inside of you. Keep asking questions and keep exploring.